Hello friends, welcome back to my YouTube channel Nest in Padasali. So today we are going to study about translational inhibitor. So this is very important with respect to CSER examination, GATE examination as well as DBT PHRF examination. So without any delay, let's get into the video. So translational inhibitors are nothing but they are the compound which inhibit the process of translation. So translation, other name for translation is that they synthesize protein. So if the protein synthesis has been stopped, then ultimately growth of cells will also become stopped. So this translation inhibitor are classified into two types. They might be an antibiotic like tetracycline erythromycin, or else they might be a toxin. Example for a toxin which inhibit the process of translation is resin. Next, these translation inhibitor may arrest the translation at different steps. So you all know translation consists of three steps. Initiation, where your ribosome will bind to a mRNA at the start codon. Next is the elongation. What do the process of elongation means? So during the process of elongation, your peptide chain will elongate by adding amino acid. And last stage is termination. So termination is a process where once the stop codon has been encountered, then all the subunits will be dismantled. So this translation inhibitor might affect the process by stopping the initiation, elongation or termination. Next, each inhibitor have different molecular target. So based on molecular target, translation inhibitor classified into translational machinery and translational factor. So if you take antibiotics like tetracycline chloramphenicol erythromycin, they inhibit the process of translation by disturbing or hijacking any process that is involved in translation machinery. Like translation machinery includes your ribosomal subunit. So they might disturb any process occurring in larger subunit of ribosome or smaller subunit of ribosome. With respect to translation factor, these factors are really important, friends. So throughout the entire process of translation, this translation factors is very important. Like IF2 is a one such translation factor. This IF2 is called initiation factor. Next, elongation factors like EFG is involved in elongation process. So without this translation factor, definitely translation will not be happen. So your examples are Puromycin and Fusidic acid. They inhibit translation by disturbing the activity of translation factor. So the next translation inhibitor might inhibit translation occurring in prokaryotic cell, eukaryotic cell or both prokaryotic cell and eukaryotic cell. So tetracycline chloramphenicol erythromycin are few translation inhibitor which inhibit the process of prokaryotic translation, whereas eukaryotic translation inhibitor include your cyclohexamide and diphtheria. Whereas if you take this puromycin and hygromycin, they inhibit translation with respect to irrespective whether the cell is prokaryote or eukaryote. They have no specificity. Next is translation inhibitor by inhibit the process both by affecting the larger subunit, like few subunits are present in larger subunit of ribosome and few process will be taking place in the smaller subunit of ribosome. So if you take this eukaryotic ribosome consists of 60A subunit and 40A subunit. So one subunit will be larger and another subunit will be smaller. So if you take cyclohexamide, which will inhibit the eukaryotic translation by disturbing the process occurring in larger subunit. Whereas if you take tetracycline, which will inhibit the prokaryotic translation by inhibiting the process that is occurring 30A subunit. So the first thing we'll study about eukaryotic translation inhibitor. The two important eukaryotic translation inhibitor include your diphtheria toxin and cyclohexamide. So first thing, diphtheria toxin, the main activity of this diphtheria toxin is that it will chemically modify this elongation factor TU. So what is the function of this elongation factor TU? This elongation factor TU act as a carrier. It's carry a charged tRNA molecule. Charged tRNA molecule is nothing but a tRNA molecule which is holding an amino acid. Just like bringing a bride to a wedding, it will also carry a charged tRNA to the A site of ribosome. So if this diphtheria toxin is present in our ribosome means, then what is the consequence means? No elongation will be taken place, definitely, because they disturb the activity of EF2. Definitely this EF2 will be non-function. As a result, it cannot able to carry a charged tRNA. Next is cyclohexamide. So the molecular target of the cyclohexamide is PTC, that is peptide transferase center, and this transferase center is located in larger subunit. Okay, what is the function of this peptide transferase center? As the name itself says, it is an enzyme and it is also a ribozyme. Okay, what does mean by ribozyme? Ribozyme means, so every enzyme is made up of protein, but this ribozyme is not made up of protein, it is made up of ribonucleotides. Okay, and this peptide transferase center is called as catalytic site of ribosome, where they covalently add amino acid via peptide bond. Okay, and what is the consequence if the cyclohexamide is present? Mean they will inhibit the peptide transferase activity. As a result, the peptide transferase enzyme will become non-functional. As a result, what will happen? They cannot able to add amino acid. Even though amino acid is present around the cell, they will not be converted to protein. Next, erythromycin, it has activity on prokaryotic cell, and the molecular target for this erythromycin is peptide exit tunnel. As a result, exit point means through which the synthesized polypeptide will be coming out of the ribosome and it will come out of the cell to participate in various functions. So erythromycin will disturb the activity of this peptide exit tunnel, which is located in 50S subunit. So what is the consequence if the erythromycin is doing such activity? Means it will block the exit of growing polypeptide chain from the ribosome. So even though the protein or the polypeptide chain has been correctly formed, this peptide chain, which is a protein, cannot able to come out because erythromycin is blocking the peptide exit channel. Okay, so the next is chloramphenicol. Again, this molecule inhibit the prokaryotic translation. The molecular target for chloramphenicol is PTC. Again, this chloramphenicol is similar activity with respect to cyclohexamide. Only difference is that cyclohexamide inhibit the eukaryotic translation, that is the molecular target for cyclohexamide is 60A subunit. Next, if you take chloramphenicol, same activity, only difference is that it will inhibit prokaryotic translation by disturbing the 50S subunit. Okay, next, what is the consequence? Ultimately, the amino acid tRNA will not be correctly positioned. So this is green color is your tRNA carrying an aniline amino acid. Since this chloramphenicol is present, it cannot able to correctly position in the A site. As a result, the translation process will not be occur. Next, Puromycin. Puromycin is really a hero. It have a different activity. As you take this chloramphenicol or other antibiotics are having different action, but this Puromycin is really a hero. First point is that Puromycin inhibit prokaryotic translation, friends. The molecular target is that it target A site and also it is near the codon anticodon interaction, and this Puromycin also inhibit translation by disturbing the process occurring in 30S subunit. So here this is the structure of tRNA. So in the anticodon loop, the anticodon molecule is present, and here this is a mRNA molecule. Okay, so here this is a ribosome involved in a process of synthesis of protein. So mRNA consists of codon and this tRNA consists of anticodon. So if the Puromycin is present, they will disturb the process of this codon and anticodon recognition, which is very important, friends. Okay, so you all know about this genetic code and all. Okay, so if the Puromycin is present and it disturb the codon pairing, means it will increase the error rate during translation. Even though the protein has been successfully synthesized and coming out of ribosome without any disturb, this protein is a useless protein. The protein doesn't code any function. By increasing the error rate, by you can able to see this frame shift mutation and all now. So just like that, it will increase the error rate. As a result, the selectivity of codon anticodon pairing will be disturbed. Next is hygromycin, same as that of Puromycin. This hygromycin also the target is present in the 30S subunit. So here 30S subunit means smaller subunit. Okay, what is the consequences? The hygromycin is present means hygromycin prevent the translocation of A site tRNA to P site. So if you see over here, one here A site, P site, E site are there. E site is a exit site and A site, P site. A site is a acceptor site and P site is a peptide transferase site. Okay, so once the one tRNA is coming in, then another tRNA should be occupy the place. Hygromycin is present, the process of translocation will be disturbed. As a result, the tRNA which is present in A site will not be moved to P site. As a result, the translation process will be freezed. Next is a tetracycline. Tetracycline also have an action only on 30S subunit. So your Puromycin, hygromycin, tetracycline have action on only on 30S subunit. So just remember PHT. Okay, it has action on 30S subunit. Sometimes in CSIR they may ask this particular question, or else it will be very very much helpful for working out some big questions asked in part C of CSIR examination. Next, consequence. So they inhibit the amino acid tRNA binding to A site. So here the tRNA is ready with having an alanine. If the tetracycline is present, it won't allow the fixing of this tRNA in the A site. As a result, the translation process will not be, elongation translation will be inhibited. Next is puromycin. This puromycin is very important, and nearly 90% of questions that has been asked in CSIR examination with respect to translation process is asked with respect to puromycin. First thing is this: puromycin is a chain terminator and it inhibits translation both in prokaryotes as well as eukaryote. And this puromycin has been isolated from streptomycin albuniger, and it is a gram-positive actinomyces. Okay, and the molecular target for this puromycin is that it will inhibit the peptide transferase center of largest subunit. Already I discussed what is the role of the peptide transferase center. Okay, now what are the consequences? It is really a difficult concept, friends. So for your notes only I had given this many points so that it will be very much helpul. So first it will bind to the largest subunit of A site, and next it's a good mimicry artist. Okay, so what it will do, it will mimic the 3' end of amino acid tRNA. So it is a mimicking agent. So actually the real person is this amino acid tRNA which is bringing the protein, but this puromycin is a fake form. It is a second copy of this amino acid tRNA. Okay, this puromycin have a modified adeno base covalently linked to tyrosine amino acid. So I'm just showing you a structure. So this is a structure of tyrosine and this is a structure of puromycin. Here you can able to see almost all the structure of tyrosine. Puromycin is having a structure of tyrosine. Okay, so these questions will be definitely asked, and this is your tyrosyl tRNA. Tyrosyl tRNA is nothing but tRNA carrying a tyrosine amino acid. Okay, so here the difference between the tyrosyl tRNA and the puromycin is that nature of bond between the amino acid and the ribosome. With respect to tyrosyl tRNA, it is a ester bond, and with respect to puromycin, it is a peptide bond. That's a major difference, friends. Okay, so here you can be able to see. So the half of the structure of puromycin is made up of tyrosine. So that's how it is mimicking the 3' end of the amino acid tRNA. So once this tRNA has been bounded to the A site, it can substitute for amino acid tRNA. So normally translation process will be going on if the amino acid tRNA has been given. But instead of this amino acid tRNA, the puromycin is occupying the place because both are having a similar structure. The ribosome cannot able to differentiate between the amino acid tRNA and puromycin. What it will do means, once they can substitute means the puromycin is very small when compared to your actual amino acid tRNA. As a result, even though it has taken up the role of this amino acid tRNA, it cannot able to synthesize the whole polypeptide chain. As a result, it will be dissociate from the ribosome as a puromycin bound peptide, and also this puromycin will cause the polypeptide synthesis to terminate prematurely. Prematurely means before the formation of full peptide itself, it will be forming a immature peptide which is of non-function, and this puromycin is acting as an acceptor of nascent polypeptide chain. Nascent polypeptide chain is nothing but growing. It's not fully grown, it's a half grown polypeptide chain which is ultimately of no group. So here is a P site, here is a site. It will occupy the A site of larger subunit. So instead of this puromycin, if some other tRNA, if actual tRNA is carrying, means then amino acids will be synthesized normally, but instead this puromycin is present, and ultimately it cannot able to take down the process. As a result, it will be dissociate with a premature termination. Okay, so you should know about this puromycylation process, friends. Similar to this amino acid tRNA, this puromycin can also able to enter into an A site of ribosome. Okay, so this puromycin is also consist of free amino acid. Okay, so this free amino acid will accept the niacin. So here you can able to see this NH2 is a free amino acid molecule present in puromycin. It will accept. Okay, but the thing is that peptide bond between the two moiety of puromycin cannot be cleaved by the incoming amino acid, because you all know the process of peptide chain elongation is done by the formation of peptide bond. Okay, so this kind of elongation will not be happen. As a result, what it will lead to irreversible premature termination of translation. This process is called as puromycylation. It is again an energy independent process. As a result, what it will lead to disassembly of ATS subunit, and also it will release the peptide puromycin product. product. Okay, so next is with respect to toxin, ricin and alpha sarsin. Actually the ricin is the actual plant protein. It is isolated from a castor point. Okay, so it is inhibit the translation occurring both in prokaryotes as well as eukaryote. The main action of this ricin is that it will chemically modify your ribosomal RNA by disturbing the process of factor binding center. So elongation factors binding center of the larger subunit. Next, this ricin is the ribosomal inactivating protein or peptide. So RIP is nothing but a large family of protein that work by acting as a rRNA N-glycolase. Okay, so this N-glycolase means what? Means they will inactivate the larger subunit by using their N-glycosidic cleavage. Okay, which releases a specific adenine base from the sugar phosphate backbone. So here you can able to see this RIP, that is ricin protein. Ricin toxin consists of N-glycosidic activity. So glycosidic activity, I think you should know about it. They will have a remove any kind of nucleotides, like if you take ricin, they cause a depurination. Depurination means since adenine is a purine molecule, they remove the adenine from GAC. What does mean by GAC? GAC means GTP associated complex. GTP associated complex is present in a place of ribosome. So inside your ribosome, this GAC is present, which is very important because for the process of translation, GTP supplies energy. So there is no ATP is involved in process of translation, only GTP is present. The ricin is present. What is the consequence? Means they will prevent the activation of translation factor GTPases enzyme. Okay, so first thing is that they will cause the adenine molecule to remove from the GAC. As a result, the GAC will not function. What will happen then? There is no GTP hydrolysis. Okay, as a result, the translation machinery will become non-functional. Okay. Next is Puromycin, and it inhibits the process of prokaryotic translation, and this Puromycin is produced by Streptomyces ramosymus, and what they will do, they will interfere with the peptide transferase reaction by acting on EFTU. Already I discussed with reference to EFTU on the first slide itself, friends. Okay, so what it will, if the Puromycin is present, then all the reactions that are associated with the EF2 will be affected, and also this Puromycin, it will not allow the EF2 to release from ribosome. What it means, if EF2 has done his activity, it has to move from the ribosome and it need to give a space for other elongation factor. Okay, so after EF2, then your EFG will be coming, which is involved in a process of translocation, that is moving your tRNA from A side to P side. Only then the peptide transfer reaction will be occurring. Okay, and this Puromycin makes your EF2 immobilization. As a result, EF2 will not be cleared. As a result, the translation process will be arrested. Next is Fusidic acid. Again, this Fusidic acid inhibit prokaryotic translation. This Fusidic acid is a steroidal antibiotic, and it is produced from a fungus called Fusidium coccineum, and it is an oral anti-staphylococcal antibiotic which is used for skin infection. These are some important point that is very important for you while working out part C question, friends. Okay, so next this Fusidic acid will bind to EFG, which is a translocation factor. So this EFG will shift the polypeptide chain from A side to the P side by a process called peptide translocation. Okay, so this EFG is really important. If the Fusidic acid is present, it will bind to this EFG GTP, which result in the inhibition of both your peptide translocation and as well as ribosomal disassembly. Ribosomal disassembly is very much important, friends, because once the peptide chain has been formed, all your ribosomes should be dismantled and it should be ready for next round of protein synthesis. Okay, and also this Fusidic acid prevents a turnover of EFG, because EFG, once it has done her duty, it has to go to home and it need to present in next cycle of protein synthesis. But if the Fusidic acid is present, it prevent the EFG to move from one place to another. As a result, the turnover will be prevented. Next is thiosteptone. These particular antibiotics are present in your James D. Watson book of molecular biology, so definitely this question will be asked for your examination. All the notes has been taken from that particular book only, friends. This thiosteptone inhibits the process of prokaryotic translation, and it is a macrocyclic thiopeptide antibiotic, and it is produced as a secondary metabolite by your actinomides. So many antibiotics are produced from this actinomides only, and this thiosteptone will also inhibit the function of this EFG, similar to your Fusidic acid. Next, the molecular target of thiosteptone is that since it inhibits the prokaryotic translation process, it inhibits the factor binding center of 50 subunit. And what is the consequence means, there is an association between the initiation factor as well as elongation factor, friends. But this thiosteptone interferes with the association of both your IF2 as well as this EFG. Okay, and what it will do means, it will inhibit the dissociation of EFG from the ribosome and also release of inorganic phosphate from EF. So generally This dioseptin will inhibit the process of GTP hydrolysis. As a result, this EFG cannot able to function. It is sitting as H. Okay. Next, so currently we discuss all the process important antibiotic that act as a translation inhibitor. Now I'll refer with the questions that has been asked in previous year question paper with respect to DBT, GATE and CSIR examination. Okay, how do most antibiotic that inhibit the protein synthesis to work? Whether they are binding to ribosome or whether they are binding to tRNA or whether they are binding to messenger RNA or whether they are binding to DNA. Okay, so definitely this DNA is a wrong option because translation DNA has no role, and messenger RNA no, tRNA no. Many antibiotic will bind to ribosome and they inhibit the protein synthesis. Okay. Next, which antibiotic is responsible for premature termination? The Puromycin. So Puromycin, there are many things, friends. You should know about the code word. This is a chain terminator and it is a good mimicry artist also, since they are mimicking the 3' end of amino. So please note, mimic the 3' end of amino acid, that is charged tRNA. Okay. Next, it acts as an acceptor for nascent polypeptide chain. This many rules are there. Next, this Puromycin has a similar role with your tyrosine amino acid. Okay, so these things definitely you should know. Next, the drug inhibit the initiation step of translation. So currently I discussed only with the elongation process, friends. What about initiation factor? So streptomycin is the only drug that will inhibit the initiation step of translation. After that, ricin, tetracycline, cyclohexamine, they have no role in initiation. They have only inhibit the process of elongation. Next, an antibiotic that resemble 3' end of charged tRNA molecule. So charged tRNA molecule and amino acid tRNA molecule both are having a same meaning only, friend. So again the answer is Puromycin. Next, this question has been asked in your DBT examination. So increasing the concentration of which of the following would most effectively antagonize? Antagonize means sorting out or coming back to normal inhibition of protein synthesis by Puromycin. So what is a Puromycin is doing? Puromycin is mimicking your 3' end of amino acid. So what are the options will be there? So if we increase the concentration of amino acid tRNA, then we can dilute the activity of Puromycin. So by increasing the amino acid tRNA to 60% or 80%, definitely this Puromycin will become inactive. So this is how the cells are coming out the activity of the Puromycin. Okay. So these are the code word that I am going to teach you, which is very much helpful with respect to exam point of view, friends. So first is diphtheria toxin DIP2. It might be your girlfriend name or your friend. DIP2. DIP2 means diphtheria toxin inhibit TU, that is elongation factor TU. Next is cyclohexamide, cyclo-PT60. Cyclo involves cyclohexamide and PT involve peptide transferase center, and 60 involve it is involved in 60S ribosome. Next is erythromycin, ERYTU50. These are tablet name, just I had given. ERY for erythromycin, TU. It inhibit the process of elongation factor TU and also they disturb 50S subunit. Okay. Next, chloramphenicol, chlorap T50. Chloro include chloramphenicol, PT include peptide transferase, and 50 includes your 50A subunit. So the cyclohexamide and chloramphenicol are the friend. Cyclo 60, chlora 50. Okay. And your erythromycin and diphtheria are friend. Diphthi TU, erythromycin TU 50. And your paromycin, paro 30, it's a hero. This paromycin inhibit the 30S ribosome, and this paromycin increase the error rate because they disturb the codon, anticodon pairing. Next, the hygromycin is also hygro 30 naughty. Hygro involves hygromycin and the 30 involves your 30S ribosome. Okay. Next thing is the tetracycline, TET30 no DT. This tetracycline is also a friend of this chloramphenicol. Okay. Next, Puromycin. Puromycin is having a cheap attitude. They are mimicking they are a second copy of your amino acyl tRNA. They mimic the 3' end of amino acyl tRNA, and also they have a similar structure similar to tyrosyl tRNA. And your ricin GT bases. What this ricin is doing? Ricin is causing a depurination of adenine molecule from GAC. As a result, this translation GT bases cannot able to work. So GTP cannot be supplied. As a result, the translation will be inhibited due to lack of energy. Chiromycin, KIT2. Chiro is a friend of diphtheria because the chiromycin is inhibit the prokaryotic translation by disturbing the elongation factor TU. That's why I am showing you KIT2. Thiostrepton is thio50G. Okay. And this fusidic acid inhibit the process of peptide translation as well as ribosome disassembly. So the next thing, prokaryotic translation inhibitor, Icmr. So prokaryotic translation inhibitor include your erythromycin, chloromycin, paromycin, tetracycline, Puromycin, thioseptine, Fusidic. Okay, it's like a Japanese word I had given. You can remember this particular thing, so you should study with the code word only, then it will be very much helpful. And next thing is your eukaryotic translation inhibitor include your DC, diphtheria cyclohexamide alone, your eukaryotic translation inhibitor. Even though if you can't able to study the prokaryotic translation inhibitor, at least study the eukaryotic translation inhibitor code word and prokaryotic and eukaryotic translation inhibitor code word. So eukaryotic translation inhibitor, DC, diphtheria and cyclohexamide, and both friends. So both prokaryotic and eukaryotic translation inhibitor include your HRP. HRP means hygromycin, ricin and puromycin. Hygromycin, ricin and Puromycin. Okay, so hope this video will be very much helpful. If you want some other content just like this particular form, then please do tell me in the comment section so that I can make a lecture for you, and I'll definitely teach you later. If I want to improve anything in my lecture, then you're always welcome, friends. Thank you. Thanks for watching this video.